19th century saw surgery transform from near butchery into effective life-saving treatment. Today, we stand at a similar turning point, at the molecular level, with the rise and refinement of Cas9 base 9 editing, the genetic surgery that could save billions.
But before we can move forward, we must first understand where we began, which would be CRISPR-Cas9 nuclease editing. We'll liken it to a butcher. CRISPR-Cas9 editing utilizes just two key components, single-guide RNA and Cas9 protein, respectively the hand and its knife. Joined as a complex, the CRISPR-Cas9 nuclease system functions as single-guide RNA directs Cas9 protein to mutations in DNA, the complementarity of their bases attracting them. The Cas9 protein then binds to a nearby PAM, a protospacer adjacent motif, and unwinds the DNA to form an editing window, where corresponding sgRNA and DNA bases can associate, like a butcher getting a good grip on the meat before he cuts it. Once these bases have aligned, chop! The Cas9 protein cleaves through both strands of DNA in what's called a double-strand break. This triggers the cell's natural DNA repair machinery, allowing the mutation to be deleted, deactivated, or corrected depending on how Cas9 cut the strands and how they're joined back together. Despite its astounding potential, CRISPR-Cas9 nuclease editing is a pretty clumsy butcher. For instance, the cleaved strands are commonly rejoined through non-homologous end joining, a process that often adds or deletes extra base pairs, sometimes producing unwanted indels and disrupting and deactivating the gene altogether. That's where we bring in the more tactful genetic surgeon, base editing. Unlike CRISPR-Cas9 editing, base editing corrects mutations by actually converting single base pairs instead of simply removing sections of DNA, requiring only a nick to a single strand rather than a riskier double-stranded break. When the good doctor gets a good grip on his patient, seeing the sgRNA and DNA base pairs line up, our surgical assistants, base editors called deanimases, can convert either cytosine to uracil or adenine to guanine. This activates the lead surgeon Cas9 nickase, a modified Cas9 protein which nicks the unaltered strand, causing the cell to see the unaltered strand as newly synthesized DNA. From there, the cell detects the new mismatched base pair and replaces the corresponding base on the nicked strand with the correct base, using the unnicked strand as a template. About half of all known pathological mutations are point mutations. It's clear to see that the stringent nature of base editing makes it the safer and more effective option. So let's treat our cleavers for scalpels as we continue to perfect CRISPR base editing, improving lives tomorrow by doing more research today.